Hello, and welcome to today's webinar organised by the Pathologist and Ultiview. I'm Olivia Gaskell, Associate Editor of the Pathologist, and I'm excited to be moderating today's online webinar where we'll review the use of 8-plex immunofluorescence staining and image analysis to gain key phenotypic data from the tumour environment. Please allow me to introduce today's speakers, Dr. Angela Vasaturo and Dr. Lorcan Sherry. Angela is a Senior Field Application Scientist at Ultiview. In 2012, she was among the first postdocs in Europe to be involved in the development of multiplex IHC and multispectral imaging and analysis of up to 6 immunofluorescent markers. She's considered a leading European expert in multiplex IHC techniques, multispectral digital pathology, and tissue imaging. Lorcan is Chief Scientific Officer at Oracle Bio. An image analysis expert, he's experienced in providing image analysis services for quantification of drug response readouts and biomarker expression within bright field and fluorescent stained histology and IHC tissue slides. He also has considerable experience in developing translational biomarker strategies. During the webinar, our speakers will present a workflow that uses an 8-plex staining protocol and detailed image analysis approach for the comprehensive assessment of the immune cell types and interactions within the tumour microenvironment. They'll be on hand to answer any questions that you may have at the end of the presentation. Please remember that you can submit questions at any time throughout the webinar. So without further ado, let's get on with today's webinar with our first presenter. Angela, over to you.
Thank you for the introduction and good afternoon, everyone. Today, together with Lorcan at Oracle Bio, I'm going to talk about how we can extract meaningful data from the tumor microenvironment by using multiplex immunofluorescence staining and image analysis. I will cover the multiplex staining component, and Lorcan will go through the image analysis component of the workflow that Ultiview and Oracle Bio have established. First of all, why multiplex? If you look at this elegant representation of the tumor microenvironment, you will immediately see how complex it is and how many different cell types are involved. Each different cell type will require one or more markers to be identified, characterized, and quantified. So it's the tumor complexity that drives the need of multiplex IHC. Although the need of multiplex IHC and the importance of multiplex IHC is now, I would say, a general consensus, the most popular staining method for routine diagnosis is still the hematoxylin and delsine, which requires the pathologist's expert eye to assess cellular and morphological features, identify the type of tissue, and also the pathological changes in a tissue. And this is actually the staining that is most effective and common procedure for the diagnosis of cancer today. Together with the H&E, and you see it in the middle side of the slide, another commonly used method in the clinical practice, today considered the gold standard, is the chromogenic IHC, like the DAB that you see in the slide. With chromogenic IHC, you are basically adding in a single protein detection, which helps identify a single cell population. However, this regular standard chromogenic IHC does not allow to assess for co-localization of markers at the single cell level. And this is because the multiple marker staining is performed on consecutive sections. And of course, this radically limits an accurate classification of cells that requires the detection of multiple markers. So from this, you immediately understand that multiplex IHC is able to overcome the limitation that I just described, because it allows the multiple markers detection and a deeper cell type characterization. Also, since we now work with fluorescence, we can add the range of expression of each of the biomarkers we are looking at, and looking at multiple cell subtypes, we can also now assess their spatial interactions. So why we are not all using multiplex fluorescence IHC routinely then? My answer to that is because the protocol is much more complex and longer, and it also needs a time-consuming development and optimization. And this is actually the challenge that Ultiview has overcome by developing a technology that has made now multiplex simple and fast. So here there's an overview of the in situplex technology. We use primary antibodies that have been conjugated to a DNA barcode, and each antibody has its unique DNA barcode. This allows us to perform just a single antigen retrieval step, which also helps keeping sample integrity. Next, we perform a single staining step that reduces the staining time and also eliminates the need for secondary antibodies, which are the main cause of non-specific binding and background signal. Next, we have the amplification step, which happens at the same time and same rate for all the antibodies and allows a broad dynamic range of expression. This increases the number of binding sites for the fluorescent probes that we will use in the next step, which is the detection step, which is based on a DNA hybridization technology. So basically, we use a mixture of fluorescent probes, which are made of a DNA sequence complementary to the barcode sequence on the antibodies, and which carries a fluorophore. We use here the classical 4 fluorophores that are spectrally distinct, which are FITC, TRITC, Cy5, and Cy7, in order to avoid spectral mixing. So this is an overview of the workflow, which now gives same-day staining to results and includes 3 different steps. So the first step is the staining step that now takes only 5 hours and a half. And with the Ultivue technology, you do not need any more assay development and optimization of your panel because Ultivue does it for you. So all the panels are ready to use. Next, we perform whole slide imaging, which depending on the tissue size can take 15-20 minutes. And then the same day, we can now move already to image analysis. And Ultivue has now established a partnership with Oracle Bio, whereby Ultivue performs the development and optimization of multiplex panels, receives the tissues from the client, and then performs the multiplex staining on those slides and the whole slide imaging, and then transfers the images to Oracle Bio, where they will be QC'd, and then there will be an algorithm developed for image analysis and data generation. But you will hear much more and more details on this topic in the next presentation. So the initial Ultivue offering has been built around T cells, due to the fact that they have a central role in the immune response to tumor, which is mediated by the interaction of T cells with other immune cells like macrophages, myeloid-derived suppressor cells, or dendritic cells, etc. We currently have available different ready-to-use multiplex kits that answer the main biological questions around cells and pathways that are crucial in responses to tumor and therefore are also targets of immunotherapy. So here you see the 6 kits that we have currently available that include T-ACT and APC kit to look at T-cell activation and professional antigen presenting cells that guide their activation, but also MDSC kit and T-RED kit to look at immune suppression in the tumor microenvironment, and PD-1 and PD-L1 kits to study T-cell exhaustion and tumor evasion. This year we have transitioned though from this fixed content to a flexible content with the generation what we call the Ultivue biomarker menu. So basically now you can choose the markers of interest in your biomarker menu and customize your kit so that you have your specific biomarkers in one kit that is optimized, and then you can apply to your tissue. Next, with the offering of the Ultivue biomarker menu and the customizable multiplex panels, you can also have now up to 8 markers stained on a single slide. So if you remember what I have shown you before of the protocol of the in situplex technology, now everything basically stays the same when you perform an 8-plex, but as you see here in this image, you still rely on one single antigen retrieval step. You will still incubate all your antibodies in one single step and then amplify all of them, all 8 antibodies at the same time and same rate. But what changes is the detection step, which now happens in 2 rounds. So you first apply a mixture of fluorescent probes that are complementary to antibody 1 to 4, and then you take an image of this 4-plex at first. Then you apply to your slide what we call the exchange technology, which basically removes your fluorescence from the sample while keeping your antibodies bound. And then you will come in with a second set of fluorescent probes that are now complementary to antibody 5 to 8. And at this point, you'll take a second image. So here is an example of what you will end up having. So you will have a round 1 image, which is a 4-plex, and then you will have a round 2 image with the second set of 4 markers, and then you will just need to overlay those to have an 8-plex 9-color image. So next, what is also important is that the initial UView biomarker menu was including all the markers that were available in the kits that I have previously described, but now we are including many more markers, so we have a growing list of relevant biomarkers that are also now not only focused on T cells, but also on other subtypes of immune cells like NKs or B cells. And we also are moving a little bit beyond that, like for example, looking at markers for cell death. What is also very important in the UView program is that we have a dedicated assay development team, which really helps you designing your project by selecting the right biomarkers to identify your phenotypes of interest. And also this team can develop new markers that you will not find yet in the biomarker menu. So if you have a marker of interest that is not yet in our menu, or if you have a proprietary antibody, we can work with it and include it in a standardized panel in the optimized, ready-to-use panel. And we can also perform validation studies like precision, accuracy, reproducibility, and eventually manufacture the kit and transfer it to you for on-site staining, or we can stain your samples in-house, or we can transfer those kits to CRO partners. So I want to give you now an example of the 8-plex that we have recently launched, which we call Ultima per IO Immuno-8 panel. And this will be also the focus on the next presentation on the analysis side. So in this kit, we have the main markers of immune cells, like T cells, for the identification of different function of those T cells with the CD3, CD4, CD8, FOXP3, and PD1, but also some markers for macrophages like CD68 and a tumor marker, pancytokeratin. So by looking at all those 8 markers, you see here a list of some of the meaningful phenotypes that you can identify and also quantify in a tissue sample. And with this, I would like to highlight also one advantage of using the Multiplex technology, which is the capability of looking at co-localizing markers on the same cell compartment. So this is an example taken from an immuno-8 stained image tissue, where you can see in white cells that express CD3, CD8, and PD1, which are the exhausted cytotoxic T cells. And it's not so common to be looking at 3 markers on the same subcellular compartment, together with a classical co-expression when you look at multiple markers on the same cell. And this is something that allows to look at what we call in-depth cell phenotyping, which you see here with some examples of the subtypes of immune cells that you can identify based on different combination of the markers that we have in this panel. Last but not least, I would like to also highlight that with those panels, and specifically I'm showing again the Immuno-8 panel, you have standardized conditions, which means that you can apply the same kit, whatever number of plexes you have and whatever number of markers you are looking at, on different tumor indications. And this is an example, and you will see it later on, the analysis on some of those tissues, with where we have applied the 8-plex on non-small cell lung cancer or colorectal cancer tissue in the same assay run. So again, showing that the conditions that we have developed are standardized and you can apply them to different indications. And with this, I get to my summary slide. I think I have shown you that we can now look at up to 8 biomarkers in a single slide by still doing whole slide imaging, that we have the ability of building custom UView kits where we can pick and mix the markers of interest. And we also have a dedicated assay development team that can develop novel or proprietary biomarkers that are not yet in our menu, and that based on the capability of looking at the expression of different markers on the same cell, we can do single cell deep phenotyping, and we can do this across multiple tissue types. And with this, I'd like to hand it over to Lorcan for the image analysis section.
Okay, thank you, Angela, and good afternoon, everyone. My name is Lorcan Sherry, and I'm Chief Scientific Officer at Oracle Bio, and I would like to talk through the analysis approach that we perform for 8-plex stained tissues. But before I move on to the detail of that, I thought I would just start by giving a brief overview of Oracle Bio for those of you that may not know us. So Oracle Bio is a specialized CRO providing quantitative digital pathology services to support pharma and biotech R&D. So essentially, we receive histology images or slides from our clients' R&D studies and use commercially available image analysis software to generate algorithms to quantify the staining. And then we return the data from those stained sections to our clients, enabling them to better interpret findings from their studies. We are a team of scientists with extensive experience of working across histology, image analysis, and also pharma R&D. And our image analysis scientists are supported by in-house clinical pathologists who perform a number of roles, such as advising on the annotation of tissue sections, supporting algorithm development and validation processes, and also ensuring that the data that we generate from our algorithms accurately reflects the underlying staining and pathology from the samples. We support quantitative pathology studies across the R&D pipeline, from target validation, preclinical, translation, and clinical studies. And all of these are performed within our quality management framework, which will also support our new GCP image analysis services coming online in January. As I mentioned, at Oracle Bio, we use commercially available image analysis software to perform the analysis, and we currently use 2 of the industry-leading software platforms, the Indica Labs platform and the Visiopharm platform. For Indica Labs, we have multiple licenses of the actual Halo image analysis platform, the Halo AI deep learning add-on, and we also have Halo Link, which is a web portal that allows our clients to log on via the web to review images and analysis that we performed on the Halo platform. Similarly, for Visiopharm, we have multiple licenses of the Visiopharm image analysis platform, the deep learning module, and we also have a proprietary Oracle Bio web portal called Observer, which, similar to Halo Link, allows clients to log on via the web to review images, analysis overlays, and data that has been performed using the Visiopharm software. Our image analysis platforms require an extensive IT infrastructure, and for that we have our GCP server is stored in a local data center called DataVita, which is a Tier 3 secure data center. But for a lot of our GCP or non-GCP and non-regulatory work, we use Amazon AWS, which allows us to store multiple licenses of the software in the cloud, but also to access multiple CPU and GPU to increase the throughput and efficiency of performing large studies. So this is an overview of the staining and image analysis workflow that Angela presented earlier. And as you can see on the right, the first 3 steps are covered mainly by UltraView with respect to the tissue sample receival, the development of the assay and the multiplex staining through to the whole slide scanning. And my presentation will focus more on the processes that happen at Oracle Bio with the images with respect to the QC of those images and pathologist annotations, the development of the algorithms, and then the processing of those algorithms to generate data across each of the images. Now, just before I get on to that process, there are a number of image analysis pre-study preparation steps that we consider and take into account before we start the study. For example, an 8-plex image can create up to 200 different phenotypes, depending on the biological relevance. So not only do you have positive cells for each of the 8 individual markers, but you also have dual cell populations, triplex, quadruple, co-localization phenotypes. And to add an added extension of that, you can also have any one of these individual markers being present in different subcellular locations, such as the nucleus, cytoplasm, or membrane. So it's important to work with the client to define upfront, where possible, the key required study readouts around the cell counts, the cell phenotypes, spatial readouts, and the regions of interest for analysis. And this really allows us to generate the algorithms as efficiently as possible. With respect to types of tissues to be analyzed, some studies can contain multiple different tumor samples and tumor types within the one study. And these different tumor types may require different algorithm approaches for tumor classification or cell formation, which again has an impact on the algorithms that we develop for the study. When it comes to choice of fluorescence labels assigned to markers, the actual order of the markers per staining round is important to reduce potential of any spectral overlap in the same cell type. Now, I know this is something that Ultivue spent a lot of time on during the assay development in ensuring that markers for a particular that are present in the same cell type are kept as far apart as possible within the order of the staining. But one of the benefits of the Ultivue technology is that the staining is done in 2 rounds. So although we've got 8 markers, they're not being forced into one staining round and then for one imaging round where we have to select fluorophores that are closer together. And of course, various studies and various tissue samples will have levels of autofluorescence and potential background staining present in the samples. Autofluorescence can mainly come in the FITC channel around things like red blood cells and extracellular matrix. And one thing we can do here is, because it's 2 rounds of staining, we can try and keep the FITC channel markers from different cell types, so that if there is areas of autofluorescence at the image analysis level, we can subtract one of the channels from the other to reduce the amount of autofluorescence present in the respective channel without actually impacting on the specific cellular staining. And this can really help in reducing or eliminating some of the autofluorescence background staining that we can see in the FITC channel. With respect to the actual images, there's different components here with respect to magnification, image file size, and file format. From a from a scan magnification perspective, normally a 20x or 20 magnification scan is suitable for detecting protein signal in cells. But 40x can be considered based on the resolution required for specific biomarker subcellular distributions. From a file size and file format perspective, as I said, 2 rounds of staining and scanning creates 2 4-plex plus DAPI images. And these 2 images can then be imported into image analysis software for co-registration. Alternatively, single TIFF layers can be created to generate a single OME TIFF image, which contains all 10 layers in the same image file. And this is actually our preferred format when working with these types of images, and it's something that Ultivue can create during the scanning process. As you may expect with 8-plex images created in the 16-bit format, these generate quite large file sizes that can be up to around about 100 gigabytes in size. So depending on the tissue and the size of the tissue, these can create big images, and it's useful and it's important to have a good IT infrastructure for storing, transfer, and working on such images. And then finally, the choice of image analysis software. There are several software packages available for multiplex staining analysis, and we currently use two, which I've mentioned, the Indica Labs Halo and the Visiopharm platform. Both have machine learning and deep learning modules to support tumor stroma segmentation. Both have modules to support multiplex cell phenotyping, and both can handle a range of file types and file sizes. From our perspective, we find it useful to be able to select a software that best supports a particular analysis approach and generation of readouts aligned to the samples that we receive. And we do this on a study-by-study basis. Alternatively, our client may request us to align the analysis we perform with the software they utilize in-house, and this can be a decision in the software we choose. For the analysis I'm showing today, we will use the Visiopharm platform. So if I move on to the actual process of the analysis, the first thing we do is we perform an image QC to evaluate each image for suitability for analysis. This involves a visual QC of each image and section, which evaluates a range of parameters such as the scan quality, any out of focus areas, tissue size, tissue integrity, any artifacts, and marker staining specificity. I've highlighted some examples of the types of things that we could pick up. So for example, small areas of tissue lifting, which can be annotated out of the image before analysis. Staining edge artifacts, so you get a sort of a brighter edge, a brighter staining at the edge of a tissue, more so on the inside, and again, this can be excluded. Or in the third example here, just an air bubble that may have crept in between two of the staining rounds, and again has impacted maybe the second round of staining. So that is something again that we can circle and exclude from the analysis. And as part of this visual QC check, we create a spreadsheet with all this information, which is passed back to the client. Marker staining specificity can be quite challenging to do in 8-plex images. Obviously, we've got 8 markers, and it can even be more challenging when you're working on a study, like a clinical study that may have 20, 50, 100 images, and then to do that across a large set of images becomes quite challenging. One of the things that we can do in this instance is we can actually export the number of pixels per marker across different intensity bin ranges per sample. And I've shown here on the right a graph where we have the 8 markers plus the 2 DAPI channels, and we have exported the pixels across a range of intensity bin ranges, as you can see on the x-axis, from the image analysis software with the number of pixels on the y-axis. So a 16-bit image has gray level pixel intensity range from 0 in black to 65,000 in white. And you can see that most of the markers here in this graph, and this is for the CRC sample, have pixel intensities between about 0 and 1800. If you look at something, if we take an example of CD4 here, you can see that there's a distribution curve for the pixels, so that the pixels peak around about 400 to 500 in the intensity range, and then they come back down, and about 90–95% of the pixels are covered within a range of 0 to 1200. So why is this useful? Well, if you're looking at numerous images across a large study set, then what you can do is if you understand the distribution for a particular marker and what the distribution looks like for good quality staining, then across a set of images you can plot the curves, and you can look for outliers indicated by certain components of the curve. For example, a skewness of the curve, which is maybe a movement of the curve to the left or right, or a kurtosis, which is a change in the sharpness of the peak of the curve. These components of the curve can actually indicate maybe outliers for that particular marker. So when you're looking across 50 samples and you have 50 CD4 curves, you can then start to identify which ones are maybe looking like outliers, go back to the image and investigate that more closely. An example here just is for the DAPI channels, and this is something that we see quite a lot for the 8-plex staining, is where maybe the second DAPI channel or the second DAPI staining is a little bit more intense than the first DAPI staining, and that's maybe to be expected because there's 2 rounds of staining, The second round of staining, the nuclei may have still retained a small amount of the DAPI from the first round of staining and imaging as part of the assay. And how that's presented here is that you can see that the DAPI1 is peaking sort of at around 300 to 400, whereas the DAPI2 in the darker blue here is peaking further to the right. And this indicates a stronger DAPI signal across the tissue for the DAPI2. This isn't necessarily an issue with respect to DAPI. We use DAPI to segment the nuclei and help, or to form the nuclei and segment the cells. And when we use DAPI, we can either use this DAPI1 channel, the DAPI2 channel, or a combination of both. But it's just an indication of how the curves can shift based on the intensities. And the overall purpose of this is to try and move away from a subjective visualization of these stain intensities, but to try and do a more objective datification of this process. So if we move on now to the pathologist annotation stage after image QC, the tumor microenvironment is annotated by pathologist to exclude any non-tumor tissue present on the section, and prominent necrotic regions are also manually excluded. I know also that after the 2 rounds of fluorescence staining and imaging, UltraView can also create a H&E section from the same sample and add that to the image, and that can also be used to assist annotations. The next step is to develop the classifier algorithm for regions of interest, and here we used a machine learning random forest classification developed in Visiopharm to segment the tumor and the stroma regions of interest within the pathologist annotated tumor microenvironment. Areas of tumor and stroma are marked up in each image, and then the pancytokeratin and the DAPI layers were used to train the algorithm. As you can see here, the tumor in orange and the stroma in blue is detected across the whole of the section for both the CRC sample and also for the NSCLC samples that we are using as part of this webinar. Moving on to the cell analysis development, this can be the most challenging part of the algorithm development phase. The tumor microenvironment contains many different cell types, and when it comes to actual cell analysis algorithm development, we see it as 2 key phases. The first phase is to identify all the different cell types that are present in the tumor microenvironment and to accurately segment those cells based on their shape and size. And then the second phase is to, once those cell objects are generated, to choose appropriate thresholds that can be applied to those cell objects to allow us to detect the individual cells and the markers and the phenotypes present within the cell objects. When it comes to the first part, which is the cell identification and segmentation, as I mentioned, the tumor microenvironment does contain a lot of different cell types. For example, macrophages, which can come in different sizes from small to large, and also different shapes with respect to either roundness or spindle-shaped cells, depending whether they're moving through the tissue. And then moving into something like T cells, which are smaller than macrophages, more rounded. They can be individual cells, or they can also be found in clusters. And then the tumor cells, which are larger cells, but much more structure to those cells and order if it's an epithelial-based cancer. And then finally, there will be all the other remaining cell types. So you can see that when we think about developing the outlines of these cells, we have to take into account the different, not just the different cell types, but their shapes, their sizes, and their distribution. And as a starting point, what we can do is we can look at the 8-plex list, and we can see that actually we have markers for each of these cell lineages, different cell lineages. So for macrophages, we have CD68, CD163. For T cells, we have CD3. For tumor cells, we have pancytokeratin. And for the remaining cells, we have the DAPI markers. So if I just walk you through an example of the algorithm development, to start with, you can see some CD68-163 staining in yellow on the screen here. And the first thing we do is we identify that CD68-163 staining based on a thresholding for what we determine to be positive and background. And then in that stain area, we perform a number of segmentation processes to, as best possible, segment this stain area into the different macrophages that are present. As you can see here, the macrophages are presenting in different sizes and shapes across the tissue. The next phase is that we look at the CD3 staining in red, and again we detect that CD3 stain area and we segment that area into the CD3 T cells. These are different to the macrophages. They're smaller, they're rounder, so we do take a different segmentation process approach for the T cells. And then similarly for the cytokeratin tumor cells, you can see the staining throughout the tumor area here in cyan, and we apply an algorithm now to segment the actual tumor cells, which is supported by the underlying DAPI present to identify the individual cells. What you can see, although the tumor cells are being formed, we are still retaining the macrophages and the immune cells present in the stroma, but also in the tumor itself throughout the tumor microenvironment. And then finally, the remaining cells where there's DAPI positive stained cells, they are picked up as the remaining cells using the DAPI layer to identify those cells. So at this stage, we have a top level layer of different cell lineages that represent macrophages, T cells, cytokeratin cells, and remaining DAPI cells. Each of these have different shapes and sizes associated with that cell type, which then allows us to move to the next phase of actually choosing our thresholds for the different underlying stains. So for example, if a macrophage has got PD-L1 present in its cell object above a certain threshold that we have chosen and defined, then that cell will change from orange to yellow, so that we end up with two populations, the macrophage PD-L1 negative cells and the macrophage PD-L1 positive cells. Similarly, for the CD3 cells, we can detect a CD8 present in the CD3 cells. This was actually in the FITC channel. And you can see some examples of CD3, CD8s in green, and they can either be PD-1 negative or PD-1 positive. And then likewise, the CD3, CD4 cells can either be FOXP3 negative or FOXP3 positive. And then we move on to the cytokeratin and we pick thresholds. The same threshold that was used for the CD68 and applied to the section is used for the cytokeratins for PD-L1, and we're able to get PD-L1 positive and negative tumor cells. And then in the remaining DAPI cells, we look for any other stains that may be of interest or cell phenotypes that may be of interest to our client. For example, they may be interested in CD8 cells that are maybe not expressing CD3. And then in that CD8 population, how many are PD-1 and positive and PD-1 negative? So from this process, we end up with 14 different phenotypes present here in this example. And then these are applied across the whole of each section within the tumor and the stroma regions of interest, both within the CRC sample and also within the NSCLC sample. And interestingly, with the NSCLC sample, we did see quite a bit more PD-L1 staining in purple. And you can see a lot of those tumor cells are now got a darker outline, but we are still retaining the cells within the tumor of the macrophages and the immune cells related to the shape of the cells that are present. So this slide here now shows some example of the quantification data that is generated per tissue type. The data generated covers everything from the total cell population counts. So for example, total CD3, total CD68, total cytokeratin positive. The example phenotype cell counts, and there's a range of different phenotypes present here. We also have some cell spatial analysis readouts. For example, we have PD-1 positive cytotoxic T cells, average distance of this cell population to PD-L1 positive tumor cells. And of course, this data is generated within each section, both within the tumor and stroma regions of interest across each section. Now, this is just a selection of the phenotypes from two sections, but you can certainly imagine that if you're performing a clinical study or a study that has tissue microarrays of up to a couple of hundred cores or something like that, then you're going to generate a lot of valuable data that can also be added to with either the patient information, the trial information, or even the therapeutic response data to enable an evaluation of proof of mechanism based on therapeutic response, or a stratification of patients based on a particular profile that the data presents. So lots of ways to mine this data to support your studies. So in summary, I have shown that a detailed image analysis approach can be used for the comprehensive assessment of key cell types and interactions within the tumor microenvironment. And the ability to carry out multi-parameter cell analysis does facilitate the delivery of key phenotypic data, thereby leading to a greater understanding of the underlying tumor biology. And of course, robust data relies on both the accurate analysis, but also high quality staining. And we believe that the partnership really supports this through the seamless interaction between the histology and image analysis phases of the study. But also, it's important that this is a client-focused collaborative workflow, and aligned communication with the client is essential. So although we've covered a lot of the technical details in this webinar, we also have assigned case study managers from each company assigned to each study. We have a kickoff meeting with the client. We agree a study plan, which covers the staining, the analysis approach, the readouts, the deliverables. And during the execution of the study, we have regular online update meetings with the client to keep them informed. And of course, at the end of the study, as well as receiving their data, our client can access all the images and the analysis overlays per sample. So to finish, I would just like to highlight that along with this webinar, Oracle Bio and Ultiview have collaborated on a couple of other studies this year. To the left, we have created ebooks. To the left, you can get access to some work that we did for a poster for the recent SITC conference in collaboration with TriStar. And this was the application of the Multiplex assay and to multi-tumor TME samples and the quantification of the data across those TME cores. And then on the right, this covers work from an earlier poster that we presented at the AACR this year, which covered the application of the Multiplex assay to colorectal cancer and NSC samples, similar to some of the work we covered today. So with that, thank you, and I will pass back to our moderator, Olivia.
Thank you to Angela and Lorcan for that insight into gaining key phenotypic data from the tumor microenvironment using Multiplex immunofluorescence staining and image analysis. We've received a number of questions from audience members, so we'll try and get through as many as we can in the time remaining, but please be sure that any questions that aren't answered live today will be answered via email after the event. Okay, so our first question is, under the present system, is it possible to image the whole slide?
And I guess that's a question for me, Angela. So yes, we always work with the whole slide imaging. I have mentioned in the beginning of my presentation that the type of fluorophores that we use are basically compatible with any type of scanner which is present today in the market, that is in the common workflows established in labs. And I think it's especially important because when we work with the tumor samples, we are aware of the heterogeneity of those samples. So we want to really avoid introducing additional biases by just selecting region of interest. And then, of course, afterwards, you can still use this entire image to annotate the tumor or the stroma, like Lorcan has shared in his presentation. And in addition to that, Lorcan also mentioned that we can perform H&E on the same exact slide where we have performed the multiplex IHC, and this also helps pathologists to annotate the samples for the analysis afterwards.
Okay, great. And how long does it take to generate a multiplex algorithm and to analyze samples?
Hi, okay, I can address that. So there are a number of steps to this process, actually. So first of all, we confirm with the client the type of and number of readouts that they require from their study. And this allows us to determine how many algorithms we need to generate and the readouts associated with each algorithm. So this can include the tissue region of interest classifier or classifier algorithms, and also cell detection and phenotyping algorithms. An UltraView will usually provide us with up to 5 images stained with the 8-plex panel, and we will use these as a starting point to generate the algorithms. And then a second, maybe larger set of images are then provided, which should represent the clinical sample types and the heterogeneity that we may expect in a clinical study. And these are used to check the algorithm cell detection, consistency, and the thresholds. The algorithm development phase can take anywhere between 2 to 4 weeks, depending on the number of algorithms and testing performed. And the client is engaged and updated through this process with online meetings and email communications. As for analyzing an image, again, this depends on the complexity of the algorithm and also the size of the tissue, but generally a large whole slide image can be analyzed within 1 to 2 hours. And we have scalable approaches from a software and IT perspective to analyze up to 8 images in parallel. So, you know, in effect, over a period of, say, 12 hours, we can analyze up to about 100 images.
Okay, great. And how long does it take to develop a custom multiplex panel?
So I have mentioned that we can basically develop also markers that are not yet in our biomarker menu in our UV program. And whenever there is a marker that's proprietary or it's a different marker that we haven't worked yet with, we require to do the assay development. And this will not take months like we are used to, but it will be just a process of 4 weeks. And for panel development, which means really putting together the marker of choice of the customer, we will take 1 to 4 weeks, depending, of course, if those are markers that are already available in our menu or if they are new markers. So we have to work a little bit more into the validation before having a full developed panel. So, but the main message here is that it's just a matter of weeks. So it could be 4 weeks for assay development and then 1 to 4 weeks for panel development.
Okay, great. And when you do multiplex, did you also do single versus multiplex? When you do multiplex, did you also do single versus multiple assays, assay concordance?
Yes, of course. This is part of our development process. We always start from DAB because, as I mentioned also during the presentation, this is the gold standard. So we always start from DAB when we work with a new antibody, and then we perform the antibody conjugation and we stain the slides with DAB with the conjugated antibody to make sure that conjugation has not affected the antibody performance. And then we move to singleplex and then to multiplex. So we really have a full standardized process to get to these validated kits.
Okay, and someone has asked, can I make a multiplex panel with markers that I have validated in IHC?
Sure, that's even a better starting point. As I mentioned, we have, we always start from DAB. So if someone has already worked with a specific antibody that works on paraffin samples and has already optimized conditions, this is a good starting point. But all the antibodies that work on paraffin samples and work with regular standard IHC can be used in our assays.
Okay, and how do you choose the readouts? And is there a maximum number of readouts that can be analysed?
Yes, I can answer that. So essentially, the choice of readouts is driven by the client that is requested the study. And what we do is we recommend the client chooses up to 20 key readouts, which are normally focused on helping to address a specific project-related question. For example, to generate evidence for therapeutic proof of mechanism, or to identify stratification biomarkers between responder and non-responder patient samples. So normally this includes total positive cells for each of the 8 markers, then around about 10 or so key dual triple phenotype readouts, followed by a couple of key cell spatial relationship readouts, but really the client can drive the components of that as they wish. We can, of course, also return mean intensity data per marker, per cell object, per image, which is exported by the analysis software. And this can be further used by the client for data mining and for further phenotypic analysis.
Okay, and this next question is for you again, Lorcan, regarding the threshold to classify cells as positive for a specific marker. Is the choice of the threshold value validated by a pathologist? How is this decision made?
Yeah, there's a couple of points here to cover. From actually choosing the thresholds, we currently perform this visually, and we use a range of samples that encompass the staining heterogeneity across the available sample set. For particular cell markers, such as things like T cell markers, macrophages, tumor cell markers, it's more straightforward to choose that threshold visually. But if you have a novel target built into a customized assay, it is helpful to have a known positive control tissue to understand both the level of the staining associated with that positive signal, and also the types of cells that are staining positive, plus their subcellular localization of the target. But as I said in my presentation, along with the visual review, we also incorporate data such as things like mean and sum of the pixel intensities per marker within tissue ROIs across image sets. And this gives us a better understanding of the variance of marker staining to make this process less subjective. Maybe just to touch upon the interaction that we have with pathologists as part of the process. Our pathologists can help us in choosing particular markers, but they also help us with respect to the validation of the algorithms. And again, this can happen at different levels. For something like a classifier algorithm, this involves performing a dice similarity coefficient evaluation, where we create fields of view across a set of the study images, and our pathologist annotates the tumor stroma present, which represents the ground truth, while our image analysis scientist independently analyzes the field of view with the classifier that was developed for the tumor stroma segmentation. And then the content of the area per ROI from each of those approaches is calculated as a dice score. A dice score is calculated from that using the dice equation, and a value of greater than 0.9 signifies good correlation between the pathologist and the algorithm. For cell analysis, again, once we have chosen thresholds and applied those, we can create field of views, and within this we can perform both precision and robustness of the cell analysis algorithms. For precision or accuracy, the field of views that we have created, our pathologist counts the number of positive cells present per marker, which again represents the ground truth, while the analyst analyzes the same field of views using the cell detection algorithm to detect the positive cells based on the thresholds that were chosen in the algorithm. And here a correlation evaluation is performed between the manual and the automated counts, and a mean correlation of greater, or an R-squared of greater than 0.9 across the image sets that have been evaluated is deemed to be an acceptable level of accuracy. And then finally, from a reproducibility perspective, we do run the algorithms over up to 5 sections, but these are we then repeat that analysis across 3 different times or 3 different days by the same analyst. And a coefficient of variance value is generated for the classifier or the cell analysis readouts to determine the robustness of the algorithm performance across those different runs. So there's certainly an input and an association with the pathologist through our selection and validation of our algorithms.
Great, thank you. And how can you run the H&E without interference between eosin and fluorescent dyes?
We are all aware of the fact that eosine have some fluorescence, give some signal. But what we do is actually we perform the H&E after the multiplex IHC staining, and we can easily do that, especially because, as I mentioned, we only perform one single antigen retrieval step. So performing H&E downstream also proves the fact that the type of protocol and assay that we are using is not having an impact on the morphology of the tissue. So, and this is the reason why pathologists can also then use this H&E to annotate the samples.
Okay, thank you. And do you validate algorithms? And if so, what do you do?
Yeah, I think this is a question I just addressed actually as part of one of the previous ones with respect to the precision, the robustness approaches, and the interaction of the pathologist to QC based on the ground truth generated from the pathologist and the analysis data generated from the algorithms.
Okay, and how do you determine thresholds in the algorithm? How do you determine thresholds in the algorithm for positive marker staining?
Yes, I think we got a question like that as well, just a few back. So again, it's working with a visual QC across a range of images, and then sort of getting that validated by the pathologist. There are other things that we should probably consider when thresholding as well. Things like background and non-specific staining across a set of images can have an impact on how the thresholds are chosen. And these can present in different ways depending on the number of study samples. Things like necrosis can have a lot of background staining associated with it. Diffuse blood cells and collagen structures can have autofluorescence. And from an assay perspective, the antibody specificity can also lead to some non-specific staining. So I know that when we work with UltraView and during UltraView's development phase of the assay, there are negative controls and positive control isotype controlled samples placed into the assay, which allow us to better understand the levels of maybe non-specific staining based on the isotype. And I also talked in my presentation about spectral overlap and the issues around autofluorescence. And there are certainly image analysis approaches that we can take regarding subtraction of signal from different image layers, especially when we have the two corresponding FITC channels present in this assay format.
Okay, thank you. And do you test for each multiplex concordance? How about steric hindrance of the antibodies?
Well, we actually completely eliminate the problem of sterical hindrance because this comes, so we eliminate the secondary antibodies, so we do not have this issue anymore. We only work with primary antibodies, basically. So we don't have this issue. And the first part, I think it was if we test each of the markers in a multiplex for concordance, and the answer to this is also yes. So when we develop a new panel, we perform the same type of validation for all the markers that are included in the panel. So for all of them, we will go through the same process, doing concordance first with DAB and then singleplex and multiplex.
Great, thank you. And someone's asked, how many slides can I stain in a workday?
Well, I would say it depends on how long you want to work in a day, but we have a fully optimized protocol to run on a Bonderix, so on an auto stainer, which means that really in 5.5 hours you can have 30 slides stained. So I would say the minimum would be 60 slides in a day because you can always run an overnight as well. If you are really a hard worker and you do 10 hours per day, you can go up to 90 slides in a workday.
Okay, and have you worked with hepatocellular carcinoma or hepatocytes? Is there an autofluorescence problem due to potential reactivity or false positive staining in cytoplasm?
Well, I have mentioned that we can basically work with any type of cancer because we have really standardized conditions that apply to any type of tissue. I have to mention, I agree, and I'm aware of the fact that hepatocellular carcinoma can have a much stronger background. Also Lorcan mentioned that, most of the times this background staining, what we call like autofluorescence, is coming into the FITC channel. We work with dyes and with an amplification method that really gives us a very high signal to noise ratio. So very rarely we experience problems like this. And in any cases, even if you have some of the background staining coming up, there is still, you can still work with it on the image analysis side. So yes, we can work with hepatocellular carcinoma as well and get the analysis performed.
Okay, thank you. And can you generate a false H from the multiplex image?
Well, this is basically what we were used to have those kind of pseudo H&E before, but now we can perform a true H&E. So really on the same slide is no longer a false or a virtual H&E. So I would say we prefer to have the H&E performed with the standard protocol on the same slide after the multiplex.
Okay, thank you. I'm afraid we're going to have to leave it there. Again, let me reassure the audience that any questions that have been left unanswered today will be answered after the event by email. This webinar will also be available on demand in just a few days' time, and you'll receive an email alert when it's ready. Thank you once again to our speakers for presenting, and thanks to everyone for joining us today. I look forward to seeing you at future webinars. Goodbye.